Welcome. To install CNVpytor, clone our GitHub repository and simply use pip to finish installation. Type CNVpytor-version to check your installation is working. After successful installation, you can import your BAM and VCF files and process them. Here, you can see a list of commands and estimated processing time for a 30x coverage sample and bin sizes between 10 to 100 KB. All imported data and calculations will be stored in a .pytor file that is now ready for visualization. Next, we'll demonstrate the visualization of data from two Pytor files, one for a blood sample and one tumor sample. To enter interactive mode, use the -view option. Interactive mode is equipped with tab completion and a help command. Just type in the chromosome name or the genomic region in order to plot it. To visualize multiple regions, separate them by a space. Set the panel parameter to add BAF likelihood panel and grid to arrange subplots. Circular plots can show RD and BAF signals in parallel, while Manhattan plots can show genome-wide RD signal. JBrowse allows you to visualize multiple data tracks at the same time that now includes CNVpytor data. Pytor file data can be exported directly from CNVpytor command line. Here is an example. It shows read depth and B allele frequency related data. We are looking for collaboration opportunities with functional annotation tools. For more information, please check the links in the description of this video. Thank you.